Okay, so we left off talking about the sickle cell anemia condition and how it relates to malaria. So they have found that the malarial environmental condition essentially will then create a situation where humans that are heterozygous for hemoglobin that have the AS genotype will have a selective advantage. They essentially will be more likely to survive because they are not subject to sickle cell anemia. They have hemoglobin that will carry plenty of oxygen to their organs, but they do have that slightly lower oxygen content in their hemoglobin that makes it impossible for the malarial parasite to finish its life cycle. So essentially what that means is if somebody with the AS genotype gets stung by a mosquito carrying malaria, they will not have the negative impact of it. So if you live in a region where malaria is endemic, the most beneficial genotype would be the heterozygous AS form. All right, so the next force of evolution we'll talk about is genetic drift. Genetic drift involves random fluctuations in gene frequencies of a population between generations. Particularly in a small population, gene frequencies do not accurately represent those of the parental population. So a good way to envision this, and we'll draw it out on the whiteboard here in a moment, but a good way to envision this is the parental population being on the mainland, and say that let's say there's 100,000 lemurs on the mainland. And let's say that only 10,000 of those lemurs are able to successfully migrate to the island of Madagascar. So those 10,000 lemurs that successfully migrated to Madagascar are not likely going to have the same genetic diversity that we saw in the parent population. And also the conditions on Madagascar are going to be quite different from the environmental conditions on the mainland. So one of the reasons that lemurs were able to successfully survive and reproduce on Madagascar is they don't really have any natural predators. Most of the natural predators existed on the mainland. And lemurs are very small primates. They're about the size of a cat or a small dog. So you can imagine if they're living on the mainland of Africa with lots of large predators, it would be pretty difficult to survive. So that's why in modern day, we have several dozen species of lemurs, but they are living mainly on Madagascar, only on Madagascar, at least naturally. Of course, we find them in zoos all over the world, but lemurs only exist naturally in Madagascar in modern day. But those lemurs went through a process of genetic drift, or what we call the founder effect. So those 10,000 lemurs, I just made up a number there, but those 10,000 lemurs that were able to isolate themselves on Madagascar were then separated genetically from the parental population. And over many generations, they eventually genetically diverged from the parent population. So those lemurs living on Madagascar were completely separate species and genetically diverged from the parent population. And in modern day, we only have lemurs living on Madagascar. So this term endogamous refers to a population in which individuals breed only with members of that population. So the lemurs on Madagascar were endogamous. They were not interbreeding with any of the lemurs that were left on the mainland. And then this term exogamous refers to a population in which individuals breed only with non-members of their population. So for example, there are some primate species that practice, essentially they will leave their natal group, essentially. Either all the males or all the females will leave their natal group at time of sexual maturation. So if all the males leave, then all the males would be breeding with females that are outside of their natal group. And then if it's female dispersal, if all the females disperse from their natal group, then all the females are mating with members of other populations. Still the same species, but other populations of that species. So exogamous, that type of breeding style or mating style, however you want to put it, definitely helps prevent inbreeding. All right, so this image here kind of gives you a visual of what I was just talking about. So the larger circle, this image here on the left, the larger circle is representing the parental or the parent population. And then let's say there's 10,000, or let's say there's 100,000 lemurs in this parental population. And then the smaller circle is representing that subset that then migrates to another geographical region. So those are those 10,000 lemurs that migrated from the mainland of Africa to the island of Madagascar. And then each subsequent circle here is representing future generations. So these lemurs are interbreeding with one another. They're isolated from the parental population. They're adaptively radiating out to fill specific roles within their environment. They're going through adaptive radiation. There's some lemurs that are nocturnal, some are active during the day, some eat mainly insects, some eat mainly tree sap. So they adaptively radiate out to fill these specific roles within their environment. And they're adapting to the circumstances that exist on Madagascar instead of the situation and environment that existed on the mainland. So this occurs when a small segment of a population becomes reproductively isolated from the larger parental population. And it results or causes a smaller population type to diverge genetically from the parental population. So eventually, genetic drift or the founder effect can result in speciation events, or essentially species diverging from the parental population. So in the image on the right, it shows the founder effect again. A few individuals from a population start a new population with a different allele frequency than the original population. So remember, allele frequencies, alleles are just versions of genes. Some of the different colors, the yellow and the red, are representing the different alleles in that population. So if by chance only the ladybugs with the red allele end up migrating to that island, then of course only that red allele will remain in the gene pool. The yellow allele will no longer exist, at least for that island population. It will still exist in the parental population over here, but it will no longer exist on the island population here. All right, genetic drift also results in a higher proportion of recessive genes in some cases, which can be lost more easily in a large population. So if in that last example, if the red allele was actually the recessive, then of course that recessive allele would be perpetuated in the island population, when maybe it might be lost or at least decreased gradually in the parental population. There's a greater chance of two recessive alleles coming together during zygote formation. So remember, zygote formation is when a sperm fertilizes the egg and produces a zygote. We get 50% of our genetic information from mom and 50% from dad. More recessively expressed traits, sometimes including genetic diseases, can occur with the founder effect and genetic drift. And also sometimes loss of genetic diversity overall because fewer individuals contribute their genes to the gene pool. All right, this image here on the right is representing the other type of genetic drift that we call the bottleneck effect. So the bottleneck effect is, again, random change in allele frequency over time and can lead to one allele being lost and the other increased in a population. And it may occur in a group that is endogamous, reproducing only within that group. So this image here on the right is showing the parental population in a bottle. And again, the different colors are representing the different alleles or variations of a gene. So in this example, we've got yellow alleles and blue alleles. And during the bottleneck event, there's a bottleneck situation where there's a drastic reduction in population size. So by chance, this could be some sort of environmental catastrophe that results in a certain portion of the population, only a certain portion surviving. Or it could be things that unfortunately are caused by humans, like the deforestation or the destruction of the environment and global warming that results in a drastic reduction in the population of polar bears, for example, or the population of mountain gorillas, or the population of cheetahs. You guys get the picture, but some sort of drastic event that reduces the population size. So by chance, what's left here in this surviving population is mainly those individuals that have the blue allele. And there's only a couple of the individuals that have that yellow allele. So then what happens, because now we have the surviving population that survived that cataclysmic event or the deforestation event or the poaching event or whatever it might have been, only the individuals, or mostly the individuals with the blue alleles, were the ones that survived. So over time, as we go through the generations, that blue allele will be increased or perpetuated within that gene pool, within the surviving gene pool. So when you look at it in comparison to the parental population, you see that the surviving or the next generation has predominantly the blue alleles, whereas the parental population had a pretty even mixture of the blue and yellow. All right, gene flow, in my opinion, is probably the easiest one to understand. Gene flow or gene migration, or you can also call it admixture of genes or movement of genes, just simply means population A interbreeding with population B, or movement of genes between populations. The ability to interbreed and produce viable fertile offspring is what defines a species. And this gene migration is also what creates cohesiveness within a species. So for example, as modern humans, especially with the advent of modern day transportation and technology, it's very possible for a human living in the United States to have a child with a human living in Japan, or for somebody in England to have a baby with somebody living in India, for example. It's not, we're not restricted to only having children with those that live in our immediate genetic, or within our immediate city or town. We can have children with people that live very far away. So this is a good thing in the name of evolution. It creates, it creates a diverse gene pool. It creates genetic cohesiveness. Modern day Homo sapiens are 99.9% identical in our genetic codes. Now, of course, we do have some variations that allow us to adapt to our environmental circumstances. And we'll talk a lot more about that actually in the human adaptation unit when we look at the variations that exist in our skin tone, our hair color, our eye form, that can all be very beautifully explained as adaptations to our local environments. Gene flow often refers to migration, though it is influenced by culture and social structure. The effects of gene flow, or the exchange of genes between populations, have increased over time, especially for humans, because we have more effective transportation with advances in technology. Planes, trains, and automobiles, we essentially have ways of transporting ourselves across the country, across the globe, and meeting humans from all different cultures and populations. So the best way to think about gene flow is simply add mixture or flow of genes, and it creates cohesiveness and genetic cohesiveness within a gene pool. So in these pictures here, we've got population 1 and population 2. Population 1 is represented mainly by the red alleles, and then population 2 is represented by the blue alleles. And we have these two populations of deer, and there's now a pass in between this mountain range. So the western deer population is now able to flow their genes or migrate their genes into the eastern deer population, and vice versa. So this flow of genes between populations, as long as these two species of deer are able to reproduce and produce viable fertile offspring, then there will begin to be a cohesiveness of traits and genetic information between the western and eastern deer population. All right, so that is the end of our forces of evolution. So now we're going to delve into what we call the Hardy-Weinberg law and the Hardy-Weinberg equation. So this is where you're going to need some scrap paper, something to write with, and a basic calculator. But first, let's talk about exactly what the equation is and what it involves. This is a method for studying genetic change within a population, because remember, evolution is essentially fluctuations in gene frequencies from generation to generation. And we know that if no change is occurring within that population, gene frequencies at a locus will remain the same. So some of that terminology from chapter 2 and chapter 3 is starting to come back. So gene frequencies, so we're going to be looking at the percentage of the dominant versus the recessive gene. We'll also be looking at the frequencies of homozygous dominant versus heterozygous versus homozygous recessive again, just like we did with Punnett squares. And that term locus simply means a location of the gene on the chromosome. So just like we did with the Punnett squares, we'll be looking at one characteristic or trait at a time. So just at earlobe form, or just at the hairline, or just at cleft chin versus no cleft chin, or just the ability to taste PTC. We're looking at just one gene, one protein at a time. And Hardy-Weinberg law will study these genotypic frequencies from generation to generation. So we know if these genotypic frequencies are fluctuating or changing, then there must be evolutionary forces operating on that trait at that time. All right, so Hardy-Weinberg law is a method for studying genetic change. So essentially, it's a method of studying evolution within populations of organisms. If no changes occurring within the population, gene frequencies at a locus will remain the same. And then if changes occurring, we know that evolution is operating on that trait at that time. So those that use the Hardy-Weinberg law would study many generations, dozens, if not hundreds of generations, to see if evolutionary change is operating on that trait at that time. So Hardy-Weinberg law will assume that none of the forces, so in order for a population to be in Hardy-Weinberg equilibrium, there must be no gene flow, no genetic drift, no natural selection, no mutation, sexual recombination must be random, or sexual selection must be random, essentially. And also the population size must be infinitely large. So those are kind of the conditions that are required in order for a population to be in what we call Hardy-Weinberg equilibrium. So that's the null hypothesis, the standard form. We need that null hypothesis to then be able to measure if there's evolutionary change occurring within that population. All right, so this slide here is very important. My recommendation is to memorize the information on this slide, but let's write it out together. The equation itself is p squared plus 2pq plus q squared equals 1. So don't let that equation intimidate you, because it actually is a very simple equation to use, as long as we memorize what each portion of it represents, and as long as we follow some very specific steps that I'm going to walk you through here in just a moment. But first, let's make sure we memorize what each portion of the equation represents. So the p variable is going to represent the frequency of the dominant allele within the breeding population. So p represents the frequency of the dominant allele within the gene pool. Q will represent the frequency of the recessive allele within the gene pool. P squared, that first part of the equation, will represent the frequency of the homozygous dominant genotype. 2pq, the middle part of the equation, will represent the frequency of the heterozygous genotype. And then q squared will represent the frequency of the homozygous recessive genotype. So one more time, because it's so important to memorize the information on the slide, the equation itself is p squared plus 2pq plus q squared will equal 1. The variable p will represent the frequency of the dominant allele within the gene pool. The q will represent the frequency of the recessive allele within the gene pool. P squared will represent the frequency of the homozygous dominant genotype. 2pq will represent the frequency of the heterozygous genotype. And q squared will represent the frequency of the homozygous recessive genotype. All right, so let's do some examples, and these are the same examples that are in your check-in assignment number 2. Check-in assignment number 2 is what's due at the end of this week by Sunday. So let's bring up our whiteboard. So for those of you that are here that are paying attention, essentially your homework will be done by the time we finish this Zoom meeting today. All right, so let's write out our equation here. So our equation is p squared plus 2pq plus q squared and bear with me, guys, I'm learning how to do these notations on the whiteboard, so bear with me here. All right, looks like there's not an option to do the little square symbol. So I'm just going to write out p squared plus 2pq plus q squared. Okay, so we always want to start with the known quantity. So if we're talking about earlobes, for example, we're talking about earlobes in humans, the dominant form is free hanging. So if you have free hanging earlobes, that means you could either be homozygous dominant or you could be heterozygous. But if you have the recessive form, which is attached earlobes, your only possibility will be homozygous recessive, like that. Okay? So that's why the homozygous recessives are the known quantity. Because if we were all in the classroom together, we could actually go around and count how many of us have free hanging earlobes versus how many of us have attached. Okay, so that's why we're always going to start with the homozygous recessive individuals within our breeding population. And hold on just 1 second. Let me pull up the actual question to make sure I'm giving you guys the actual question for this one. To pull up our check-in assignment number 2. So if anybody else wants to also pull it up on your computer, we're within module 2. And then we're down underneath the tab called Demonstrate, which is check-in assignment number 2, the Hardy-Weinberg equation. Let me just look at the actual question here. So our first one. All right. Oh, so the earlobe one is the second one. So we're actually going to be doing question 10 first. So the allele for free-hanging earlobes, uppercase E, is dominant over the allele for attached earlobes. In a population of 10,000 individuals, 2,500 will have attached earlobes. So we're going to determine the frequency of the dominant and the recessive alleles, and then we're going to plug in those values into the Hardy-Weinberg equation. So ultimately, we're going to be answering the question, what percentage of generation 1 would be homozygous dominant for earlobe form? So we're going to follow these steps exactly to calculate genotypic frequencies. We're going to take the number of homozygous recessive individuals and divide by the total number of individuals within the population. So let me write those steps down here for you. All right, so we're taking the number of homozygous recessive individuals and dividing by the total number of individuals within the breeding population, because that is going to give us our Q squared. So Q squared. So the number of homozygous recessive individuals is 2,500. And we're going to divide that by the total number of individuals within our breeding population, which in this case is 10,000. Sorry, guys, bear with me. This text box thing. So it's a Q squared will equal 2,500 divided by 10,000. Because we're taking the number of homozygous recessive individuals and dividing by the total in the population. Okay, so 2,500 divided by 10,000 is going to give us our value of Q squared. We know that Q squared is going to equal 0.25. But careful, that's our value of Q squared, and we need the value of just Q. So the opposite in mathematics, the opposite of squared is the square root. So on your computer, or on your calculators, or on your computer calculator, whatever you're using, it's the one that looks like a little check mark with a line across the top. So it's going to look like, let's see if I can draw it for you. It's going to look like this. That's the square root symbol there. So when we take the square root of 0.25, we're going to get the value of just Q. So everybody do that in your calculators. Take the square root of 0.25, and that'll give us our value of just the variable Q, which is representing the recessive alleles in the population. We know that Q equals 0.50, and we always go out to 2 decimal places. So now we have our value of Q. So the way we got there, we took the number of homozygous recessive individuals in the breeding population, which is 2,500, and then we divided it by the total number of individuals in the population, which was 10,000. So 2,500 divided by 10,000 gave us 0.25, but we want to get down to the value of just Q. So the opposite, or the reverse or inverse of Q squared, would be the square root of that number. So we took the square root of 0.25 to give us 0.50. So if we know that Q equals 0.50, it's actually really easy to find the value of P. because we know that the dominant alleles plus the recessive alleles in a population have to equal back to 1, or 100% of the population. So P plus Q must equal 1. And we know the value of Q, so all we need to do is 1 minus the value of Q, which is 0.50, will give us the value of P. So we take 1 minus 0.50 will give us P. So we do the math, and we know that P also equals 0.50. Okay. All right, so now that's essentially the conclusion of step 1 and step 2, because we know now that P, the variable P, or the dominant alleles, represents how the percentage or the frequency of dominant alleles within the breeding population. And we know that P equals 0.50. And we know that Q, which represents the recessive alleles, we know that Q also equals 0.50. Okay, so that's the conclusion of essentially step 1 of the Hardy-Weinberg equation. Step 1 involves finding the value of P and finding the value of Q. All right, so then let's erase this stuff so that we can do step 2. So step 2 of the Hardy-Weinberg equation will involve plugging everything in, plugging in those variables or the value of those variables into our equation. So the equation itself, P squared plus 2PQ plus Q squared is going to equal 1. All right. So when I say plug it in, all I mean is that whenever we see a variable, we want to plug in the value of that variable. And that's the information that we obtained through the process of step 1. So whenever we see a P, we're going to plug in the value of P. So P is 0.50 and it's squared. So squared just means multiplying it by itself. So 0.50 multiplied by 0.50. So that's the first part of the equation there. That's the P squared. The second part, the 2PQ. So we're going to do a 2 multiplied by the value of P, which is 0.50, multiplied by the value of Q, which is 0.50. So there's the middle part of our equation there, the 2PQ. And then the last part of our equation, the Q squared. So we take the value of Q, which is 0.50, and multiply it by itself, 0.50, is going to hopefully get us back to 1. All right, so there we have it all set up. So the first half of our equation is right here. 0.50 times 0.50, that's P squared, which will represent the homozygous dominant genotype. The middle part of the equation, 2 multiplied by the value of P multiplied by the value of Q will give us the frequency of the heterozygous genotype. And then the last part, 0.50 multiplied by 0.50, that's the Q squared portion, which will give us the frequency of the homozygous recessive genotype. All right, so everybody in your calculator, 0.50 multiplied by 0.50, that's going to give us 0.25, and that's going to represent the homozygous dominant portion of the population. It's going to look like that. If we wanted that number there, the decimal, we call that a frequency. If we want to turn a frequency into a percentage piece of cake, all we need to do is multiply it by 100 or simply move the decimal place 2 positions. So 0.250, we move our decimal 2 places to the right, and that becomes 25%. So we know that 25% of this breeding population is homozygous dominant for earlobe form. And then middle part of the equation, we punched into our calculators, 2 multiplied by 0.50 multiplied by 0.50 will give us 0.50. And that's representing the frequency of the heterozygous genotype. Okay, so 0.50. And then if we want to turn that into a percentage, also a piece of cake, we just move our decimal 2 places to the right. So 0.50 becomes 50%. We know that 50% of our breeding population is heterozygous for earlobe form. Last part of our equation, the 2p cubed portion, or excuse me, the q squared portion. So the 0.50 multiplied by 0.50 again gives us 0.25. And that'll be the frequency of the homozygous recessive individuals in the breeding population. So to make it a percentage, 0.25 becomes 25%. So we know that 25% of our breeding population is homozygous recessive for earlobes. So nice thing about the Hardy-Weinberg equation, you can also add together the frequencies and make sure you get back to 1, and you can add together the percentages and make sure you get back to 100% of the population. So everybody in your calculator, 0.25 plus 0.50 plus 0.25 gets us back to 1. We know for a fact that it does that. So that gets us back to 1. And we also know that 25% plus 50% plus 25% gets us back to 100% of our breeding population. All right, so all the Hardy-Weinberg problems that I give you, you know for a fact that they will always, the frequencies will always equal 1, and the percentages will always equal 100% of the breeding population. All right, so that was practice problem, or that was question number 10 within check-in assignment number 2. So now we're going to go back and do question number 9. So we're going to follow those steps again exactly. You guys have any questions so far? All right, so let me pull it up again just to see what question we're looking at. Okay. All right, so we're still on check-in assignment number 2. Now we're doing question number 9. So the allele for dimples, uppercase D, is dominant. In a population of 1,000 individuals, 200 individuals have no dimples. So that means 200 individuals are homozygous recessive. So we're going to determine the frequency of the dominant and the recessive alleles, and we're going to determine what percentage of the population would be heterozygous for dimples. So we're going to follow the same exact steps. So remember, we're always beginning with the homozygous recessive individuals, because for dimples, if you have dimples, that's the dominant phenotype. So you could either be homozygous dominant or you could be heterozygous if you do in fact have dimples. If you do not have dimples, the only way you would have no dimples, you would need to be homozygous recessive. You would need a copy of the recessive allele from both mom and dad. So that's why we're beginning with the homozygous recessive individuals. That's the known quantity, because if we were actually looking at a population, we could count the number of individuals with dimples versus the number of individuals without dimples. And those without dimples, we know that their genotype must be homozygous recessive. So that's why we're beginning with the known quantity. All right, and we know that that's Q squared. So Q squared will equal 200 because that's the number of homozygous recessive individuals. And we're dividing that by the total number in the population, which is 1,000. So we're going to find our value of Q squared by taking 200 and dividing by 1,000. So everyone do that in your calculators. 200 divided by 1,000 to find the value of Q squared.
That's 5.
So when I put it in my calculator, 200 divided by 1,000 equals this number here. Is that what you guys are getting?
Yes.
Okay. So Q squared equals 0.20. But careful, this is usually the step that students forget. So be very careful not to forget this step, because that's our value of Q squared. And what we want right now is a value of just Q. So the inverse, or the opposite of squared, is the square root. So we need to hit that symbol that looks kind of like a check mark. Whoop, sorry guys. Let me see if I can make that a little bit better. There's not a whole lot of control on this drawing tool. So it looks kind of like a little check mark like this, and then a line along the top. Okay, so that's the square root symbol there. So everybody hit that square root symbol in their calculators. And that will give us a value of just Q. So somebody tell me what's the value? What's that?
It's supposed to take 6.
Excuse me?
Huh? You can't be addicted to CMOS.
Oh,
Okay, so guys, remember, if you're not asking a question, just make sure you're on mute. That way we're not hearing your background noise. Okay, so you guys tell me, what is 0.20? And then hit the square root button. What's our value of Q rounded out to 2 decimal places?
I got 0.45.
Beautiful, perfect. So our value of Q is 0.45. Okay, and now that we have our value of Q, and we know that the number or the percentage or frequency of the dominant alleles plus the frequency of the recessive alleles will have to equal 1. So if we know that, and we have the value of Q, it's easy, piece of cake, to find the value of P. We just take 1 minus the value of Q, which is 0.45, and that's going to give us the value of P. So everyone, in your calculators, what's 1 minus 0.45?
0.55.
Beautiful job. All right, so we know that P equals 0.55. All right, so that's the conclusion of step 1. We have our value for P. We know that the dominant alleles, the frequency of the dominant alleles is 0.55. And we know that the frequency of the recessive alleles is 0.45. Okay, so my advice, once you get to the conclusion of step 1, definitely circle your value of P and Q, and be careful not to mix them up. Okay, we need to be very careful that we don't mix up our values of P and Q. All right, so let's go ahead and erase that information so that we can now do essentially step 2, or part 2 of the Hardy-Weinberg equation, which will involve plugging in the values of P and Q into our equation. All right, and my advice is always, always, always set it up this way to avoid any confusion. So our equation itself is P squared plus 2PQ plus Q squared will get us back to 1. All right, so there's our equation. And then when I say plug it in, essentially what I mean is anytime we see the variable P, we're going to plug in the value of P. So in this case, it's 0.55. So we're doing the P squared portion right now. So it's 0.55 multiplied by 0.55. And then the middle part of our equation, the 2PQ. So we're going to take 2, multiply it by the value of P, which is 0.55, multiply it by the value of Q, which is 0.45. So there's a middle part of our equation there. And then the last part of our equation, the Q squared. So we're going to take the value of Q, which is 0.45, multiply it by the value of Q, which is 0.45. And hopefully that's going to get us back to 1. All right, so here's where your calculators come in. Everybody in your calculators, what is 0.55 multiplied by 0.55, rounding your answer out to 2 decimal places?
0.30.
0.30, and that's representing which genotype? P squared represents the
Homozygous dominant.
excellent homozygous dominant. We're talking dimples here. So we're going to do 2 capital letter Ds. All right, so if we wanted to turn that frequency, that 0.30, into a percentage, how would I do that?
Two decimal points to the right.
Exactly. We just move our decimal 2 places to the right. So 0.30 becomes 30%. So we know that 30% of our breeding population is homozygous dominant for dimples. All right, the middle part, the 2PQ. So everybody in your calculators put in 2 multiplied by 0.55 multiplied by 0.45, and then round your answer out to 2 decimal places.
Point 0.50.
Beautiful. 0.50. And that's representing which genotype?
Heterozygous.
Heterozygous. So 1 uppercase D and 1 lowercase. If we want to turn that into a percentage, what would it be?
50%.
50%. So we know that 50% of our breeding population is heterozygous for dimples. All right, last part of our equation, the Q squared portion. So in your calculator, 0.45 multiplied by 0.45, and then round your answer out to 2 decimal places.
120.
0.20, and that's representing homozygous recessive individuals, which would be into a percentage would be 20% homozygous recessive. Okay, so everybody in your calculators, just to make sure we get back to 1, what's 0.30 plus 0.50 plus 0.20? Do we get back to 1?
Yes, you get back to 1.
Beautiful. We get right back to 1. And then also 30% plus 50% plus 20. We get right back to 100%.
100.
Or 100% of that breeding population. All right, so my advice, guys, is always, always, always set it up like this so that you can see, so you can follow it down. So you can see the P squared portion. You can see the frequency and the percentage. You can see the heterozygous portion, the 2PQ, the frequency and the percentage. And then you can see the Q squared, the frequency and the percentage. And you want to make sure the frequencies always, always add up to 1. And the percentages always, always add up to 100%. Okay, you always want to follow those steps exactly. All right, how you guys doing? You doing okay so far?
Yep.
All right, so let's do the next practice problem.
That was question. That
All
was
right, so
number 9. That was question number 9 we did.
correct. The one we just did was question number 9, because we actually accidentally did 10 first. We did 9 just a moment ago. We did 10 first. And now we're going to do question 11 on check-in assignment number 2. So this one is the third Hardy-Weinberg practice problem. And this one says the ability to taste the chemical PTC is controlled by a dominant allele, uppercase T. In a population of 5,000 individuals, it was determined that 750 of the individuals were unable to taste PTC. Determine the frequency of the dominant and recessive alleles. All right, so I'm going to do step 1 first. So remember, we're always going to start with the known quantity. So in this situation, if you're a taster, that's the dominant condition. So if you're a taster, you're either homozygous dominant or you're heterozygous. But if you're a non-taster, that's the recessive condition. If you're unable to taste PTC, your only possibility for your genotype is homozygous recessive, little t, little t. Okay, so that's why that's the known quantity, because if this was an actual population, we could actually use tasting papers and determine how many of us can taste PTC and how many cannot. And we would be able to actually know that those non-tasters are homozygous recessive. So that's why we're always starting with the known quantity, which is the number of homozygous recessives divided by the total number of individuals in the breeding population. And that's going to be our value for Q squared. So in this case, it's 750 divided by 5,000. All right, so you guys tell me with your calculators, what's 750 divided by 5,000?
.15.
Beautiful. So we know that Q equals 0.15, or Q squared, excuse me, Q squared equals 0.15. Okay, now careful, this is usually the step that students forget because that's the value of Q squared, and what we want is the value of just Q. So what do I need to do to find the value of just Q?
Square root 0.15.
Beautiful. So we take that square root. And please excuse my horrible drawing abilities on this whiteboard. But we're taking the square root of 0.15, and then we're going to round that answer to 2 decimal places. And that'll give us the value of just Q. So what's the square root of 0.15 rounded to 2 decimal places?
.02.
I'm getting something different.
Oh, point 39, sorry.
Beautiful. Nice job, guys. So our value of Q equals 0.39, like that. And we know that the number or the frequency of the dominant alleles plus the frequency of the recessive alleles will have to equal 1. And we already know the value of P, or excuse me, we know the value of Q, so it's easy to find the value of P. You take 1 minus the value of Q, which is 0.39 in this case, is going to give us a value of P. So you guys tell me, what's 1 minus 0.39?
.61.
Beautiful, 0.61. All right, perfect. That's the end of step 1. We now have our numerical frequency for the value of P, or the dominant alleles within the breeding population. And we also have a numerical value or a frequency for the Q variable, or the recessive alleles in the population. So my advice, once you get to the end of step 1, make sure you highlight or circle or put a box around. Make it obvious to yourself what is the value of P and what is the value of Q before you move on to step 2. All right, so let's go ahead and erase this information so that we can do step 2. So remember, step 2 involves plugging in the numerical values of P and Q into the Hardy-Weinberg equation. So the first portion of the equation, the P squared, so we're going to take the value of P, which is 0.61, and we're going to square it or multiply it by itself, 0.61. And then the middle part of the equation will be the 2PQ. So we're going to take 2, multiply it by the value of P, 0.61, multiply it by the value of Q, which is 0.39. Okay, and then the last part of the equation, the Q squared. So we're taking 0.39 and multiplying it by 0.39. Sorry, I kind of ran off the side a little bit there, but you guys see it. All right. Okay, so then we just plug it in into our calculator. So you guys tell me, what's 0.61 multiplied by 0.61 and then rounded to 2 decimal places?
0.37.
0.37. Oops. All right, 0.37, and that's representing which genotype?
Homozygous dominant.
Homozygous dominant, so big T in this case. If we want to turn 0.37 into a percentage, how would we do that?
Move over 2 decimal places.
Exactly, move our decimal over 2 places to the right, so that becomes 37%. We know that 37% of our population is homozygous dominant for tasting. All right, then the middle part of our equation, 2 multiplied by 0.61 multiplied by 0.39. What's our answer rounded out to 2?
0.48.
0.48. And that's representing which genotype?
Heterozygous.
Heterozygous, so big T, little t. How do we turn that into a percentage? 0.48 becomes
48%.
48%. So we know that 48% of our breeding population is heterozygous for tasting. And then the last part of our equation, 0.39 multiplied by 0.39. What's your answer rounded out to 2 decimals?
.15.
Beautiful, 0.15. And that's representing which genotype?
Homozygous recessive.
Homozygous recessive, beautiful. And then if we want to turn that into a percentage, what percentage would that be?
15.
15%. So we know that 15% of our breeding population is homozygous recessive for tasting. All right, so last thing we want to do, we want to make sure that all equals, the frequencies need to equal 1 and the percentages need to equal 100%. What's 0.37 plus 0.48 plus 0.15? Do we get back to 1?
Yep.
Yep, we do. And 37% plus 48% plus 15%, we get back to 100% of the breeding population. Okay, so the question itself at the very end is asking what percentage of generation 1 would be heterozygous for the ability to taste PTC? So our answer would be 48%. All right, so I'm going to have you guys go into your breakout rooms to do the last one. And I'm going to put the problem in the chat box here. So everyone take a moment and write this problem down. So a small group of 100 people decide to isolate themselves from the world and move to a small and remote deserted island. Out of this population, 10 of the individuals have albino skin, which is due to a homozygous recessive condition, little a, little a. All right, so let's put you guys into some breakout rooms. So really what you need to write down is the number of homozygous recessive individuals, which is 10, and then the total number of individuals in the population, which is 100. All right, let's do 5 breakout rooms. All right, so in room 1, I have Kevin, Felix, Jose, Jocelyn, and Victoria. Room 2, I have Alex, Lauren, Lily, Lisandro, and Luis. Room 3, I have Alejandro, Jacob, Kaya, and William. Room 4, Anthony, Francisco, Karen, and Natalie. And room 5, I have Damien, Gustavo, Leslie, and Paul. So let's go ahead and take about 10 minutes in our breakout room. So do this practice problem and also discuss if you have any further questions for me about chapter 4. And I'm going to broadcast this message to you guys too, just in case you need it.